Bismillahirrahmanirrahim. We start another paper 5 too, and in this I'm putting in some tips to help you solve the paper 5 in the upcoming exam very soon. First of all, the point which is not clear to many of you is what is the independent variable and what is the dependent variable. This is a very favorite question. It's a 2 marks question, and anybody losing those 2 marks would be a very sad day for me. So independent variable is something which you are going to change. I independent, which I am going to change. Are you going to change what? In this situation, in this picture that you've seen, we are applying the fertilizer X in this experiment. So that is the independent variable. What are we measuring? We are measuring the result of it is the dependent variable, and that is the variable affected by the change in the independent. So if you put more fertilizer, more growth, more number of leaves, more number of fruits. So what are you going to measure? You're going to measure the number of leaves. So you're going to apply a fertilizer. Maybe you apply it for a month or you apply it for 6 weeks, and then you're going to record the number of leaves in the beginning and then the number of leaves after 6 weeks or 8 weeks. So what are you measuring? What is the result? I always say dependent variable is the result. What are you measuring? You're measuring the number of leaves. So the number of leaves is the dependent variable, not that you are measuring growth. The growth is a process which you are measuring. Like for instance, you're measuring the rate of photosynthesis, but how are you measuring it? You're measuring it by the number of bubbles produced per unit time. So you're measuring, you're not going to tell me what process you are measuring. You're going to tell me how you are measuring it. If you're going to say, okay, I'm measuring the rate of photosynthesis, that's incorrect. You're measuring the number of bubbles produced per unit time. Independent variable is what you have changed. I have changed. I is the independent variable. Another picture to show you that the dependent variable responds to or depends on the independent variable. That's another very nice way of saying it. So the amount of light you receive is the cause. You can control it. You can increase or decrease the light intensity. You can increase or decrease the light intensity. And this independent variable is always plotted on the x-axis. That is why I always say the mnemonic for this is FIX. You see, if you have a table, in the first column, you always put the independent variable. So light intensity would go here. And this was always plotted on the x-axis. The dependent variable is the effect. How are you going to measure? The length of the stem or the number of leaves, or you're going to measure it in some way, you're going to measure it. And the dependent variable is always plotted on the y-axis. Another very simple way I want to suggest to you all is to remember is, does sunlight affect the growth of a plant? So dependent variable which we are measuring is the growth of the plant. How we are going to measure is the length of the stem maybe. Is the stem going to be longer or is it not going to grow at all? Independent variable is sunlight. So we're going to expose it to the sun. One will be exposed more to the sun, will be exposed less to the sun. So what are we going to control? The other variables which we are going to control is the amount of water should be kept the same. We use the same type of plant. We use the same potting mix. The soil which is in the potted plant, it should be the same. And we use the same sized pot in which we have got the soil, in which we have the nitrates and the phosphates and all that which is going to affect the growth. So light is just one thing which you are going to change. So that what you change is the independent variable. That what you measure is the number of leaves or the length of the stem is the dependent variable. Now another example for how we want to word it, an experiment testing the effect of different amounts of fertilizer on the plant height. So plant height is a dependent variable and different amounts of fertilizer is what you are changing is the independent variable. So I am changing this. Plant height is the dependent variable, which is the result. Ask what must stay the same to be fair. So same fertilizer brand, same type of plant. You can't be testing it on corn and then testing it on rice and then saying, okay, no, I'll test it on wheat or I'll test it on sugarcane. It has to be the same crop. So same fertilizer brand, same measurement standard. Are you going to measure the length of the stem? Are you going to use a ruler? Are you going to use a tape? And the same amount of water and exposure to light, the timing and the light intensity. How long is it going to be exposed to light? 7 hours, 8 hours, 10 hours. What is going to be the wattage of the bulb? Is it going to, if you're going to do this in the lab, is it going to be the 100-watt bulb? Is it going to be an 80 wattage bulb? So all the other things must remain constant, because what are you changing is you're changing just the amount of fertilizer. So the mass of the fertilizer applied, is it 1 gram in 100 ml, then the mass and the volume must be the same. The concentration of the fertilizer must be the same if you are making it out of some sort of a powder and then dissolving it in water. Now, as we all know, this is on the same day as paper 2, so your paper 2 and paper 5 is going to be on the same day, and this is for 1 hour 15 minutes. Now, you've got to understand this is a 1 hour 15 minutes, but this is only and only for 30 marks. Now, if you look at your paper 2, the paper 2 is also for 1 hour and 15 minutes, but that is for 60 marks. So please remember your paper 5 is only for 30 marks. But why is the same time given to you? The reason is it's a very, very thinking paper. You need to think a lot of things. You need to read the question at least 3 times. You need to understand the question. Most of the answers are in the question, as I will tell you in this paper when I solve it for you. Most of the answers are in the question, but if you have read the question carefully and you have underlined the keywords and you have made a note of it, the second thing which you've got to remember is you've got to do it as if you were doing it in the lab. So in your mind, you must pretend as if you're doing in the lab. And then, of course, all those mark scheme points will come back to you and you'll be able to do a good job of it. Now, coming to the first question of this paper, which is the March 2023 paper, which is the latest paper. Potatoes are underground organs made by the potato plant, Solarium tuberosum. Figure 1.1 shows a potato plant. So you can see the normal leaves, there's the stem, there's the upper part, and in the soil we have these potatoes growing in it. And they would initially be small, and then slowly they would grow bigger and bigger and bigger, and then we would have eventually harvested. So it is actually the potato size which is increasing. So look at the picture carefully, visualize it, explain it to yourself. Okay, so that means the leaves are photosynthesizing, glucose is being made here, and then the glucose is converted into sucrose, and then it's going to go here, and then it's going to be stored in the potato as starch. And the potatoes are going to get bigger and bigger and bigger and bigger. And then, of course, we harvest it and then we go and sell it in the market. Potatoes contain high quantities of starch and are a popular food. After harvesting, potatoes are stored. During storage, the starch content decreases. So if it was 100 gram starch in the beginning, after storing it for 1 month, maybe it's gone down to 80 grams. Do this in your mind. During storage, the starch content of potatoes gradually decreases due to the breakdown of starch molecules into glucose. So you had 100 grams of starch, and maybe after 4 weeks now there is 80 grams of starch. What has happened is that some of the starch has been converted to glucose and the glucose has been respired. So starch was initially 100 grams, and after storage, it says during storage the starch content gradually decreases during storage. So you've kept it for, well, just think, I mean, we are just imagining it. We've kept it for 4 weeks and we measured the starch in the beginning and then we measured it at the end, and we find out that starch has decreased. A glucose assay, assay means a measurement. A glucose assay using a Benedict's test and a colorimeter can be used to determine the concentration of glucose in potatoes. Now, this we've never heard of this before, but it's a new situation, and this is what they want to see, how a student is able to comprehend a new situation and then answer the questions on it. It's actually testing your very important concepts in biology and how clear you are on that. I think this paper is amazing, how it checks your understanding of the subject or the topics. The glucose assay uses these steps. The outer skin is removed from the potato. Imagine you're doing this. The potato is cut into small pieces that are then ground into a pulp using a mortar and a pestle. Now you can see this is a mortar and pestle. Now look at the difference. Number one, look at the difference in shape. Number two, look at the difference in the material. Number three, look at the pestle. One is made of wood, one is made of some sort of a rock, and one is made of some sort of a ceramic thing. So this is a mortar and a pestle. Now, if you are not very good in visualizing things, well, you're going to not be able to do this paper. So you must be able to visualize all this, which I'm telling you now. So the potato pulp is filtered. So we made this potato pulp. The potato pulp is filtered to remove most of the solids and obtain potato juice. So we've got a juice now here. We've removed the particles which might have been in it or the solid parts in it. Excess Benedict solution is added to the potato juice. So if you took say 5 ml of the potato juice, you added 10 ml of the Benedicts. Mentally now you're going doing this. You're doing to do this exactly in the paper how I'm doing this. So excess Benedict solution is added to the potato juice. So I said 5 ml potato juice, 10 ml Benedict solution. Why? Because it's the copper in it which reacts. So if it has more glucose and you've added less copper, well, it's not going to react. Say it had 100 glucose and the Benedicts that you've had has only got 50 copper. So you don't know what is the glucose content of the potato juice. So you add more of the Benedict's solution. The mixture is heated in a water bath at 90 degrees Celsius for 5 minutes. During this 5 minutes period, copper ions in the Benedicts react with the glucose in the potato juice to form an insoluble precipitate. The mixture is then filtered to remove the precipitate. The filtrate, which is blue, contains copper ions that did not react with the glucose in the potato juice while the mixture was being heated. So, you know Benedict's is light blue in color. So we've got Benedict solution light blue in color. Then we've got another Benedict solution which is dark blue in color. And then we have another Benedict solution which is very dark blue in color. I can't really get that very dark blue. Now, what you have to understand is it says the mixture is filtered to remove the precipitate. The filtrate, which is blue, contains copper ions. So this one contains 50 copper ions. This one contains 100 copper ions. This one contains 150 copper ions. Why? The filtrate, which is blue, contains copper ions that did not react with glucose. So the more the glucose, the less the copper ions. Now here is the inverse story which they expect you to understand. The filtrate, which is blue, contains copper ions that did not react with glucose. So I gave you these figures 50, 100 and 150. So that means only 50 copper was left. The rest of it all reacted with the glucose. But in this, because there's 100 copper, it means that there was less glucose. So 100 copper did not react. This one has 150 copper. So that means even this had even lesser glucose. So the filtrate, which is blue, contains copper ions that did not react with the glucose in the potato juice while the mixture was being heated. A sample of the blue filtrate is transferred to a colorimeter tube, which is called a cuvette. A colorimeter is used to measure the absorbance of the blue filtrate. A calibration curve is used to determine the glucose concentration of the potato juice from the measurement of absorbance. So now I want you to start thinking. Light blue, mid blue, dark blue. Light blue, mid blue, dark blue. The darker the color, more the absorbance. So if this is dark blue, that means it has less glucose in it. Because more the copper means that there was less glucose. So this is what you have to understand. A colorimeter is used to measure the absorbance of the blue filtrate. So you have three situations, a light blue, a mid blue and a dark blue. In the light blue, we have 50 copper. In the dark blue, we have, mid blue, we have 100 copper. In that, we have 150 copper. So more the copper means less the glucose. Figure 1.2 shows one type of colorimeter. Hole to hold the colorimeter tube. Digital readout of absorbance. Colorimeter tube, 3 centimeter tube of sample to be tested, which is the blue filtrate. A clean colorimeter tube containing the sample to be tested is placed in the colorimeter. Light is passed through the sample and the absorbance of light by the sample is measured. Outline how colorimeters should be prepared before carrying out measurements on samples so that correct absorbance readings in the glucose assay are obtained. So somehow you've got to standardize it. So number one, you have to ensure that the correct color or the wavelength of light. Either you said that, or you said use a colorimeter tube which contains just distilled water or deionized water or distilled water, and that is the zero absorbance. That is the zero absorbance. So you set it at zero absorbance, when you put in a tube containing just distilled water is not going to contain anything else in it. So distilled water, or if you use the word deionized water, so either you could have said use of distilled water, or you could have said the correct wavelength of light. To determine the concentration of glucose in potato juice using the glucose assay, a calibration curve needs to be produced. This involves using the glucose assay to obtain absorbance reading for standard glucose solutions of known concentrations. A student was given a 2.0% stock solution of glucose from which to prepare a range of standard glucose solutions of known concentrations. State the glucose concentrations the student could use to produce a calibration curve and describe how 20 cm cube of each solution should be prepared by proportional dilution of the 2%, 2.0% stock solution of glucose. You may use a table to show your answer, glucose concentrations to use, how to prepare. Now you have to understand that there is a difference between serial dilution and proportional dilution. Serial dilution is when you have an original stock solution of 2 glucose. So now you've taken 5 centimeter out of this and you put it 5. Now you can see it here. I'm just going to color this. So 5 centimeters going into here, and then 5 centimeter distilled water is being added. Then 5 centimeter from here. So now this would be 1 From 2 now this is 1 And then from 1 it is 0.5 then it is 0.25 and then it is 0.125 So 5 centimeter of one goes into the other, and then 5 from here goes into other, and then you add another 5 of distilled water to it, then 5 from here. So serial means that you do it in a series of test tubes. So about 5 test tubes are lying there, and you take from one and put it into the next, and then from that you take it to the next and next to the next. So from A to B, B to C, C to D, D to E. So this is called serial dilution. Now this is proportional dilution. Proportional dilution means that you had a 1% standard solution. Now you're going to make a 0.8%. So what did you do? You took 8 cm cube of this original solution, 1%, and you added 2 cm of water into it. Then in this one you have added now 6 cm. I'm just going to put an arrow to it. You've added 6 cm here, and you've added 4 cm of water to it. And then in this one you have added 2 cm, and then you've added 8. So totally all of them have 10 cm cube of fluid in it. This is a 1%. This is a 0.8%. This is a 0.6%. This is a 0.2%. So you have changed the solution from 1 to 0.8 to 0.6 to 0.2. Well, you could have had 0.6 to 0.4 and then 0.2. Now in this solution, we have 1% standard solution. Now we are making a 0.1. So you've only taken 1 cm and you've added 9 cm of water. So from 1%, this has become 0.1%, and then it's gone 0.01%, and then 0.001%. And each time what you've done is you've added 9 cm water and you've transferred 1 cm each time. So this is now, of course, a serial dilution. This is not a proportional dilution. This was a proportional dilution. Now, as you can see, this is something which we also check in your paper 3, which if you've taken the exam of paper 3, which was in the AS level, some of you will have done that. So we've done this in paper 3, and this is for 3 marks. For 3 marks, all you have to do was give me this table. It says 5 stated concentration and units. So I decided to take 2.0% glucose, 1.5% one. Now you always write the units in the headings. You don't write it in each of the lower columns. Then how much stock solution are you going to have? You had a stock solution of 2.0 stock solution. So you only had 2.0%. So it has to be all less than 2.0%. You can't make a 3% solution. No, not from a 2%. You can't evaporate the water from it. So from 2.0, 1.5, 1.0, 0.5, and 0.0. So I've got 5. It says 5 stated concentration and units. And then reasonably evenly spaced. You could have said 2 1.8 and then 1.5 and then 1.2 and then 1 or zero point 0.5, something like this. You should be evenly spaced. And then you gave me the method for dilution, that you would add 20 cm cube of the stock solution and then you would add 0 distilled water. In 1.5 you'd add 15 of the stock and 5 say cm cube of distilled water, because you had to have a total of 20 cm cube. The main thing you've got to understand is that this is again the point is that you have to read the question carefully. So the total had to be 20 cm cube. If you look at a calibration curve, well, this is what it's going to look like. So more the glucose concentration means more, so less copper in it. Less copper means it's going to be, so you see a negative correlation. More the concentration, less the absorbance. Now coming to the B part of the question, the student decided to investigate the effect of storage time on the glucose concentration of potatoes. You are measuring, identify the dependent variable, you are measuring the glucose concentration, but basically you are measuring the absorbance. So glucose concentration of potato measured as the absorbance. So you got a mark for that. The student was provided with freshly harvested potatoes, standard laboratory apparatus, and a colorimeter. Describe a method using the glucose assay that the student could use to investigate the effect of storage time on the glucose concentration of potatoes. The method should be set out in a logical order and be detailed enough to allow another person to follow it. Details of how to carry out the glucose assay and how to prepare and use the colorimeter should not be included. Now, some of you did not read this note, and you gave me all that. Same potatoes. We mean the growing conditions or the variety of potato or the harvesting time of the potato. So we mean that. Same potatoes, we mean that. I want you to understand that. The second point was that you would store them in the same conditions. They must be all at, say, 10 degrees Celsius or 6 degrees Celsius. The fridge is at 4 degrees Celsius. So some temperature, and they would be in a dark place, and it would be a dry conditions, not humid. It should be low humidity. Some additional information about this was how did you keep them? Storage conditions, sealed bags at 5 degrees Celsius in an air conditioner set on no humidity, or dry air. Then storage time, you must give me at least 5. Like I always say, if you are talking about temperatures, give 5. Anything else which you are varying, the independent variable which you are changing is storage time. So 4 to 6, 8, 10, 12 weeks, or you could have even said this in months. So of course that would be decided by the examiner what would be acceptable at this point. Then same volume of potato juice. Same volume of potato juice. So at each storage time, measure the absorbance of the potato juice. Then I use at least 3 measurements for each storage time, or you can say use 3 different potatoes for each storage time. And then you calculate a mean. Then this is a medium risk experiment. So you have to give me the safety, hazard, and precaution. So it's a medium risk experiment. In the potatoes, you could be allergic to it, just like I'm allergic to mangoes. Use gloves. Then Benedict's reagent is an irritant. Use gloves. So you could have come up with any of the points which are relevant to that very question. When potatoes are cooked at high temperatures to make potato chips, glucose and amino acids react together. This reaction is known as the Maillard reaction and is responsible for the orange-brown color of potato chips. When glucose reacts with amino acid asparagine, acrylamide is made. So asparagine plus glucose, high temperature cooking at more than 120 degrees Celsius results in acrylamide. The reaction is shown in figure 1.1. Low concentration of acrylamide. So low concentration of acrylamide, say 10, are produced when potatoes are cooked at high temperatures to make potato chips. High concentration, 100 of acrylamide, are toxic to humans and can increase the risk of certain cancers developing. So less acrylamide formed at high temperatures to make potato chips, and high concentrations are toxic to humans. Exposing raw potatoes to gamma radiation decreases the acrylamide concentration of potato chips. Some scientists investigated the effect of gamma radiation on the composition of raw potatoes. 4 bags of one variety of potato were each exposed to a different dose of gamma radiation, 0 J kilogram, 50 joules kilogram, 100 joules per kilogram, and 150 joules per kilogram. This is the gamma radiation that we're giving them. 3 samples of raw potatoes from each bag were then analyzed chemically to determine the concentration of glucose, protein, and volatile nitrogen compounds. 3 things: glucose, protein, and volatile nitrogen compounds as a percentage of dry mass. Volatile nitrogen compounds are present in potatoes due to the breakdown of some proteins and amino acids such as asparagine. Statistical tests were carried out on the data to assess whether the gamma radiation dose affected the concentration. So when the gamma radiation was less or when it was more, does it affect the concentration of glucose, protein, and volatile nitrogen compounds in the raw potatoes? Data from the chemical analysis and statistical tests are shown in table 1.1. So we have the gamma radiation dose 0, glucose is 0.83, protein is 10.92, and volatile nitrogen compounds is 1.14, 10 raised to the power minus 2. 50, this, 0.86, 11, 100, 0.83, 10.0. You must do this. You must read it very carefully. You must see a trend. So as the gamma radiation dose is increasing, the glucose is sort of increasing. 83, then it's 88. And the mean composition of the raw potato sample as a percentage of the dry mass. The protein is nearly the same, 10.92, 11.04, 10.08, 10.96. Not much difference, but there is marginally. But the volatile nitrogen compounds are decreasing: 1.14, 0.75, 0.68, and 0.40. Statistical significance of differences between raw exposed is not significant, less than 0.5, not significant. This is significant at 0.05. The scientists suggested that gamma radiation reduces the acrylamide content of potato chips by affecting asparagine. Explain how the information provided and the data in table 1.1 support this view. Now, what we have to understand is that we have to look at from the data and from the question. As gamma radiation increases, volatile nitrogen compounds decrease significantly. So you see this decrease. Now, this was in the question. There was nothing you coming up with something brilliant ideas. It's just the things of the question that you were writing. Then no change in percentage of glucose. So there's no change in the percentage of glucose. Therefore, decrease in acrylamide is not due to glucose. And the third point which you could have said is that decrease in volatile nitrogen compound indicates decrease in asparagine, so less acrylamide is made in the Maillard reaction, which was again in the question, the Maillard reaction. Gamma radiation increases volatile nitrogen compound, decrease significantly, no change in percentage glucose, therefore decrease in acrylamide not due to glucose, decrease in asparagine, so less acrylamide made in the Maillard reaction.